The final talk in the first half of the session this morning will be by Jessica Deere.
Hi everyone, and thank you to everyone online and here in person for joining my talk on how social interactions influence disease transmission in wild primates. So in social animals, social conditions shape the environment that individuals experience from day to day, and social adversity is closely linked to health and mortality outcomes. We see here that in many social mammal species, morbidity and mortality decreases as social advantage increase. So I don't need to tell anyone here that sociality is important, but we all very well know that sociality also brings disease. So while we know that social bonds are an integral part of group life and can benefit health, there's also a cost of sociality in wild animals through the increased risk of pathogen transmission through proximity and contact. A better understanding of the role group living plays in disease transmission is important, particularly in animal populations with heterogeneous or fluid social structures, such as chimpanzees. Chimpanzees live in multi-male, multi-female communities characterized by fission-fusion dynamics in which the size and composition of their groups varies throughout the day and also from day to day. So this social dynamic provides a good opportunity for investigating this relationship between sociality and transmission of disease, including respiratory diseases, which, as we can see by the publications here, are widely recognized as a threat to endangered chimpanzees and other great apes. These diseases often originate from human populations, but patterns of transmission within wild populations are not fully understood. But we do know that respiratory diseases have the potential to cause a significant decline in populations. Outbreaks with mortality rates up to 19% and morbidity rates as high as 100% have been reported in communities of wild great apes. So understanding respiratory disease transmission in chimpanzees is crucial to their health, especially in populations habituated to human presence for research or tourism. And you can see in this picture here that chimpanzees are going about their termite fishing business while tourists observe, and they don't mind at all. So direct sampling of throat or nasal swabs would be the standard for diagnosing specific respiratory pathogens in humans. But these materials cannot be obtained from wild animals without immobilization and anesthesia, which are associated with risks to animal safety and have negative effects on behavioral research. So we need to be able to collect samples non-invasively. And while the number of validated diagnostic tests for non-invasive samples, like fecal samples, is growing, there are still logistical and financial constraints for deploying these methods or employing these methods at a large scale. So syndromic surveillance, which is based on the collection of pre-diagnosis health data, is a non-invasive method for disease monitoring. And can be used or can be recorded using a health sheet like this, in which you observe health data, including respiratory data. You can record the signs and frequency of coughing, sneezing, and runny nose, along with other health parameters. But an inherent assumption of syndromic surveillance is that observed clusters of disease are epidemiologically linked, which would ideally be confirmed with diagnostic tests. But when diagnostic data are unavailable, a contact network approach may be utilized to explore epidemiological links among observations of syndromic respiratory disease. So social network analysis is a method of mapping and measuring contact patterns within a network of individuals, and can be used to examine how social contact influences disease transmission in wildlife populations. A network consists of nodes, the dots or individuals, and edges, which are the connections between the individuals. And network metrics are used to calculate the importance of nodes for transmission or potential transmission pathways throughout the network. And syndromic surveillance and social network analysis are particularly useful in study systems where individuals are already undergoing observation for behavioral research, which is the case in Gombe National Park, where the chimpanzee population has been studied for more than 50 years. Gombe National Park is a small forest reserve located on a narrow strip of land in western Tanzania, in between Lake Tanganyika and a rift escarpment that rises from the lakeshore. It is currently home to 3 communities of chimpanzees and several other primate species. And many of these primate species are habituated to human presence and live in close proximity to humans. So indirect, non-human primate and human interaction is common, as you can see in this picture here, with chimpanzees and baboons together in an area where humans live and congregate frequently. So the presence of researchers, park staff, and tourists provides many conservation benefits, but also provides disease transmission risks. So we investigated whether social network analysis can provide insights into the seasonal incidence of syndromic respiratory disease in wild animals. The aim of the study was to evaluate the contact networks of wild primates in association with patterns of syndromic respiratory disease using social network analysis tools and long-term health and behavioral data from a community of wild chimpanzees living in Gombe National Park from 2005 to 2016. This study included 95 individuals, 56 males and 39 females. And the chimpanzee was considered positive for syndromic disease if they were observed to have signs of coughing, sneezing, or running nose during a given season. We quantified 3 different network metrics for association and grooming contact. Degree strength, which tells us about the total number of connections the focal node has weighted by the frequency or duration of adjacent connections. Betweenness, which measures the number of shortest paths the node has between 2 other nodes. And then closeness, which is an estimation of how closely connected the node is to all other nodes in the network. And additionally, because possible transmission pathways in the population may not be adequately represented by the observed network, we applied the network K-test and the weighted version, which is path-based K-test, to determine whether the network has epidemiological relevance. And we explored this relationship between syndromic respiratory disease and social contact using binomial generalized linear mixed models. We ran 3 different models, 1 for each metric, each with association and grooming contact, season, age, sex, and time observed as fixed effects, and a nested random effect with individual and sample size. And we calculated the association index or association contact using a dyadic association index, which is the proportion of 15-minute interval focal follows of 2 individuals in which they're together in the same group. And the grooming contact is the proportion of time 2 individuals spent grooming with each other when they're both together in the same group and one of the individuals is the focal follow. So here is the graph of incidence of syndromic respiratory disease by season. You can see seasonal incidence along the y-axis and the 12 study years along the x-axis from 2005 to 2016. And the 4 seasons, early and late wet season and early and late dry season, are represented by the different colors. Incidence ranged from 0 to 84% depending on the season. And you can see where some potential outbreaks occurred with higher rates of syndromic disease, like the late dry season of 2011. So neither degree strength nor betweenness had a significant relationship with incidence of syndromic respiratory disease. So here I'm showing the results of the closeness model. Odds of having clinical signs of respiratory disease in individuals with higher closeness were increased by 1.36 in the association contact network and 1.28 in the grooming contact network. And odds of syndromic disease in the late dry season were high, or compared to the early wet season, were also significantly higher. These are representations of networks from 2 of the seasons that had epidemic levels of syndromic disease, which was defined as at least 20% of the population being infected. These are also the seasons that had significant K-test results. So the distribution of cases within the networks in these seasons was consistent with dissemination through network links based on the path-based K-test, which demonstrates that observed contact networks in these seasons are important in explaining transmission during disease outbreaks. However, this wasn't the case when nearly the entire population was infected, and this is likely because we would need to use a finer time scale to see patterns of transmission at extremely high epidemic levels. But the path-based K-test can provide additional support of infectious outbreaks in the absence of diagnostic data to confirm pathogen presence. So to conclude, closeness is important for reflecting an individual's ability to spread infection to the entire group. So its significance indicates that individuals' position in the network is important in the potential spread of respiratory disease. And this is consistent with other studies that have shown that global metrics often play a more important role than local metrics in understanding the spread of directly transmitted infections, whereas local metrics might be more important when environment plays a role in transmission. Sorry, I'm speeding up because I think we're getting close on time. But so this was the case when, in a similar study we did in the same system around the same time period, and we found that a local metric, degree strength, was important in the study of environmentally transmitted gastrointestinal parasites, whereas closeness was not. So the pattern, we did see that the pattern of syndromic respiratory cases was consistent with transmission of a pathogen through contact networks during 2 of the seasons with epidemic levels of syndromic respiratory disease. And given this observed spread of syndromic disease in the system, our findings stress the need for further respiratory disease diagnostics to gain a better understanding of the transmission pathways of respiratory diseases in wild primates, which is what we're currently working on at Gombe Stream Research Center, where we recently expanded molecular lab capacity at Gombe and trained the Gombe veterinarian and lab technician on conventional PCR methods, so that way we can perform real-time diagnostics and learn more about disease transmission dynamics in a system with human-wildlife overlap. So I want to make sure to acknowledge the many, many people and teams involved to make this possible, and especially the field researchers who go out every day, rain or shine. Thank you.
Thank you, Jessica. Yeah, we've got time for maybe one question.
Sure.
So that was really fascinating.
Thank you.
So respiratory disease is kind of generic. Do you know what, is it paramyxo? Do you know what the actual etiological agents are?
So for Gombe, we don't know yet, but we're currently just about to launch screening for 5 respiratory viruses, so we'll hopefully know soon. Some of them are 4 respiratory viruses and a bacteria, strep pneumoniae. So we're going to start screening for common wild chimpanzee pathogens. So strep pneumoniae, RSV, metapneumonia virus, COVID, SARS-CoV-2, and one other. Yeah, so we don't know yet, but we'll hopefully know soon.
Excellent.
Thanks.
Thank you, Jessica.
Thank you.